Hi there, welcome to AB Bio Fun with Dr. D. And today we're going to start talking about the cell division cycle, cell division, mitosis. So, the cell theory, one of the tenets of cell theory is that all cells arise from only pre existing cells. So, how is that possible? How is that accomplished? Well, it is accomplished through cell division. So, cell division is needed for reproduction, and this is true mostly for single celled organisms, except some multicellular organisms reproduce asexually. It is also needed for growth and development of multicellular organisms. And of course, it's needed for repair and regeneration of tissues in multicellular organisms. Multicellular organisms need to coordinate cell division across different tissues and organs because if they don't, the result is usually cancer. So, this is critical for normal growth and development and for maintenance. It is critical that multicellular organisms coordinate the timing of cell division. They also coordinate the rates of cell division. Not all cells can have the same cell cycle, and that's very important to understand. So the frequency of cell division really varies by cell types. If we talk about small prokaryotic organisms such as bacteria usually divide every 20 minutes. Now let's talk about us, multicellular organisms. A human embryo cell cycle is less than 20 minutes. That means every 20 minutes, a cell will divide. That is because the embryo needs to grow very fast. Our skin cells are also cells that tend to divide frequently throughout their life because we end up shedding a lot of skin. They divide usually on a 12 to 24-hour cycle. We have liver cells that retain, some of our liver cells retain the ability to divide, but they're kept in reserve and they really divide only once every year or two. And finally, mature nerve and muscle cells do not divide at all after maturity. They're permanently in G0. And we're going to explore later what G0 means. So the cell cycle phases are as follows. We have S, which stands for synthesis. This is where DNA replication occurs. And we have M, which stands for mitosis. Mitosis includes nuclear division and cytokinesis. Cytokinesis is the division of the rest of the cell. Prior to S phase, we have the gap phases. G1 stands for gap 1, and G2 stands for gap 2. So G1 is prior to S phase, and G2 is prior to M. So they're the gap phases between mitosis and S phase. And because originally scientists only knew about S phase and mitosis, these phases were called gaps. So that's the gap between M and S is G1, and the gap between S and M is called G2. G1, S, and G2 are collectively called interphase. So this slide basically summarizes all the major events that are happening in each of the phases. In G1, cells grow, and the cellular contents, excluding the chromosomes, so everything except for DNA, gets duplicated. In S phase, it is the DNA that gets duplicated. Each of the 46 chromosomes we have is duplicated by the cell. In G2, the cell will double check that everything is okay. It'll make sure that all the DNA is replicated, all the chromosomes are replicated, there are no errors, and everything is okay and everything is ready for mitosis, because mitosis is now nuclear division, and cytokinesis will be the rest of the cell being divided. Now, the logic of the mitotic cell division cycle is as follows. The goal is the faithful duplication of the genetic material. That means the mother and daughter cells need to be genetically identical. And this goal is achieved through highly ordered sequences of events and feed forward and feedback links and loops. And I already mentioned in our last video on cellular communication and feedback regulation that feedback loops are everywhere, and feedback loops are used quite frequently in cell cycle regulation. Now, what does that mean? It means that an event is dependent on the correct completion of a preceding event. An event also determines the timing and the nature of the following events, and an event can negatively influence a preceding event, ensuring that a forward movement and occurrence of an event only once per cell cycle. For instance, we want to be able to duplicate our DNA only once. And if you remember this model, I use this model for feedback regulation to show you how metabolic pathways use negative feedback regulation. This model can be used also to explain the cell cycle. So the first event is event A. Event B is dependent on event A being completed. Event B can never happen without event A. Event C cannot happen if event B and A have not happened. And event D happens only after A, B, and C have happened. So event D is dependent on the completion and correct timing of the preceding events, A, B, and C. Event D can also inhibit prior events. This ensures that the regulation, the direction in which the events are moving is only one way. So you go from A to B, from B to C, and from C to D, and you can't go back to A. You can't do A, B, C, A, B, D, B, A, B, C, D. It has to be A, B, C, and D. And this is assured by positive feedback loops and negative feedback loops, showing that events are dependent on each other. So let's explore a little bit in detail what is happening in the different phases. S, as I said, stands for synthesis, and synthesis means putting things together. This is the phase of the cell cycle where cells will duplicate mid-exact second copy of all of their chromosomes. And one chromosome really is one large molecule of DNA. So this is where DNA replication will take place. After S phase is completed, S phase is followed by gap 2, the gap phase. This is the phase where the cell will pause and execute checkpoints. And checkpoints are points where the cells will pause and make sure everything that has occurred is completed and is completed accurately. So let's say event B checks that event A is done, it's completed, and is completed accurately. In this case, the event would be DNA replication. So in G2, the cell will check whether DNA is completely replicated, whether there are any errors during DNA replication that need to be repaired. And it will check sure that it has everything else in place, like the spindle, and it has grown to the right size, and it's ready really to divide. Now, I left G1 last because G1, the gap phase one, the first gap phase, is actually the most important phase of the cell cycle, the most important phase and interphase. It occurs right after mitosis and prior to the next S phase. And that's the phase that couples cell growth to cell division. So a cell needs to reach a certain growth, a certain size before it can divide. And of course, when a cell reaches a size which is too large, then it needs to divide. We explored that in our unit 2. Gap phase 1 or G1 is the most important phase in a life's cycle because it is the major control and it's the major decision point in a cell's life. So a cell can be viewed, the life of a cell can be viewed as a shifting balance between mitogenic means divide, and anti-mitogenic, that means stop, do not divide, signals that it receives. And at any point, a cell can receive multiple signals from its environment. Remember cellular communication. Cells receive signals from their environment. The signals are either from the environment, but they could also be internal signals. And most signaling pathways actually that involve grow, that tell the cell to grow and divide, the mitogenic signals, are kinase signaling pathways, the ones we explored, for example, the EGF receptor. So signals that tell the cells to grow and divide function mostly through these receptor tyrosine kinases. Anti-mitogenic signals. So mitogenic signals are very simple. The ligand binds and the signal that the cell receives is grow and divide. That's it. Now, anti-mitogenic signals basically tell the cell stop, do not divide. But they can be very different. They could be go ahead and die, apoptosis, apoptotic signal. They could be listen, starvation. You don't have enough food. You don't have enough energy, enough resources. You shouldn't divide. They could be contact inhibition. We explored this in juxtacrine signaling, if you remember. This is how we maintain tissue integrity, right? If cells are surrounded, if a cell is surrounded on all of its sides by cells, by other cells in a tissue, that would be a signal not to divide. And of course, there's signals that make cells become specialized, differentiate. So there's signals that signal the cells to differentiate. All of these signals are stop, do not divide. They're anti-mitogenic. Mitogenic, the word mitogenic comes from the words mitosis, divide, and genic, generate divisions. So let's explore a little bit the G1 phase. The G1 phase has an important control point called the restriction point, or it's often also referred to as the G1 checkpoint. It's a checkpoint because this is where the cell checks all the information it has received and makes a decision. And the decision, once it's made, it cannot be reversed. So the cell weighs in all the signals, both internal and external, both the signals that it's getting from the environment as well as sensing its own state. And if the signals that tell the cells to divide outweigh the signals that tell the cell to stop, if you can see here at the bottom, mitogenic signals outweigh anti-mitogenic signal, the cell will proceed past this checkpoint and will actively start dividing. Once the cell proceeds past this checkpoint, there's no turning back. Even if something goes wrong, this cell is committed to cell division. It's committed to mitosis. Now, if the signals that tell the cell not to divide, the anti-mitogenic signals, that are the opposite, don't divide, outweigh, they're more than the mitogenic signal, then the cell will not divide. It will adopt a different fate. And it depends what the signals are. It could undergo apoptosis, it could differentiate, or it could just retire. Senescence is another term for a cell being, I'm too old to be dividing. I'm just going to sit here still and do my thing. But all of these decisions are made right here in the G1 checkpoint. And this is a very, very important control point in the life of a cell. All right, let's now explore mitosis. We explored the phases of interphase. Now we're going to explore the phases of mitosis. And most of this should be a review for you. All right. So G2 of interphase, we have in this model, what you will see the nucleus with the nuclear envelope. You will see the nucleolus, the plasma membrane, and you will see the centrosomes with the pairs of centrioles, right? The chromosomes are duplicated because DNA replication has just been completed, but they're uncondensed. In prophase, the chromosomes will condense and they're visible. If you look under the microscope, you can actually see them. And the early mitotic spindle begins to form. That means that the two centrioles will start moving apart from each other and the spindle fibers are going to start coming out of them. In prometaphase, the spindles are beginning to attach to the kinetochore. Kinetochore, the two duplicated chromosomes are called sister chromatids. They're attached kind of at the center by a special structure called the kinetochore. And the spindles will attach to that structure. In metaphase, these spindle fibers, they attach like hooks to the kinetochore of the sister chromatids. they will pull the sister chromatids in opposite directions. This spindle will pull towards itself and this spindle will pull towards itself. But the sister chromatids do not separate because there is a glue that holds them together. So on the metaphase plate, this is where the sister chromatids align. They align in the middle of the cell. And they align in the middle of the cell because the spindle fibers are pulling them in opposite directions. Yet the two sister chromatids are held together by a molecular glue. In anaphase, the molecular glue that holds together the sister chromatids is broken down and the fibers are pulling the sister chromatids in opposite directions. And by the way, I did forget to mention that in prophase, the nuclear envelope, the nuclear membrane, will begin to disintegrate, to fall apart. So the nuclear membrane actually disappears. In prometaphase, there's no longer a nuclear membrane. In metaphase, there's no longer a nuclear membrane. So in anaphase, the sister chromatids are separated, are beginning to get separated. In telophase, which is the final stage of mitosis and which is followed by cytokinesis, the sister chromatids are now called chromosomes, and they're already pulled in opposite directions and the opposite ends of the cell. And the cell will start to pinch in the middle. This is called the cleavage furrow. So it'll initiate separation of the remainder of the cell, the cytoplasm and all of its content. And the nuclear envelope will start reforming. Okay. And these were just models. And now I'm just going to show you beautiful microscopic pictures of what cells look like. In G2 phase, and in blue you will see DNA, and yellowish red, it's actually yellow, but when it combines with red, it comes out to kind of greenish, are the fibers. So in prophase, if you look hard enough, you will see already like squiggly worms. These are the chromosomes, so the chromosomes have condensed. In prometaphase, you can see the spindles in green. They appear like kind of greenish yellowish here. And you see that they're kind of already at the opposite end of the cell, and the spindle fibers are protruding and attaching to the sister chromatids. In metaphase, in blue, you will see the sister chromatids, all the DNA is kind of in the middle of the cell, and the spindle fibers are emanating from them. Anaphase, you'll already see how they're beginning to separate. They're being pulled apart. Telophase and cytokinesis, you will see how the blue is already in opposite ends, and you will see how kind of in the middle the cell starts to pinch. This is the cleavage furrow. All right, to be continued. Remember, biology is the only science where multiplication and division mean the same thing. We'll talk more about cell cycle in the next series of lectures.